In this section, we're going to talk about erythrocytes, which are the actual red blood cells. So remember, this is the densest part of the blood, and it's about 44%-ish of the volume of your whole blood. Now, there are a ton of erythrocytes. They are very, very small molecules, and there are about 4.2 to 6.2 million per cubic millimeter. So what does that mean? Okay, well, a cubic millimeter is the same as 1 microliter, which is 1,000th of a milliliter, which is 1,000,000th of a liter. Okay, so there are 1 million cubic micromillimeters in a liter. On average, there are about 5.2 million erythrocytes per cubic millimeter. And normal human blood volume, there's an asterisk here, I will come back to this, is about 5 liters. So if we take this average of 5.2 million erythrocytes per microliter times 5 million microliters in the volume of blood in your typical human, that gives us 26 million erythrocytes, or 26, oh my gosh, so many zeros in the average human body. All right, let me talk about this asterisk here, average and 5 liters of blood. The average human, for anatomical and physiological measurements, is a white male of European descent. So this average human is not actually the average human that exists on the planet. The average human, the most common human on the planet, is actually an Asian female who actually has a lower blood volume because she is a smaller human being. Of course, the actual blood volume depends on the actual size of the human. So some people who are very small might have 3.5 liters of blood. Some people who are very large might have 6 liters of blood. So 5 liters is a nice rounded number, but remember that it is not actually indicative of any normal human being. Also, yes, for the purposes of physiological and medical research, the average human is a white male. Females are abnormal humans, according to this definition. Anyone who is not a white male is an abnormal human, according to this definition. Yes, that is hella racist and sexist. I'll come back to that later. All right, let's go back to our erythrocytes. Erythrocytes are adorable tiny little cells. They are not even a full micrometer across, so you could fit more than 1 million of them in a meter. They're biconcave, meaning that the top and the bottom are both concave. They're little round cells smushed in the middle, kind of like if you took a jelly donut and smushed all of the jelly out of the middle, you would get something sort of shaped like a red blood cell. This is really cool because they can bend as they go through capillaries, which is where gas exchange happens, and there's tons of surface area on this cell that can push up against the walls of the capillary and promote gas exchange. Within each red blood cell, there are about 280 million hemoglobin molecules. The hemoglobin molecule looks like this. And these little lines in here are showing all of the hemoglobin molecules within this cell. The hemoglobin molecule is the thing that actually binds to the oxygen molecule. And remember, we're talking about O2, oxygen gas, okay? So this is the hemoglobin are actually carrying the oxygen gas inside the red blood cells. Now, hemoglobin itself is a 4-part molecule. It is a protein made up of 4 protein subunits, and we call these globin chains. In the adult, there are 2 alpha globin chains and 2 beta globin chains, and they're slightly different, not in a way that we're going to worry about in this class. You'll learn more about that in physiology. The hemoglobin of a fetus actually has different chains. It still has these 2 alpha globin chains, but instead of beta globin, it has gamma globin. And because of the structure of those gamma globin chains, the fetal hemoglobin actually has a greater affinity for oxygen than adult hemoglobin, which is why as blood passes through the capillaries in the placenta of the mother, the oxygen will flow down a concentration gradient into the blood of the fetus in the fetal capillaries, so that the fetus always takes oxygen from the mother. After birth, these hemoglobin molecules are going to be destroyed. The fetus is going to make, or the baby at that point, is going to make new red blood cells with new hemoglobin and replace the gamma globin with these beta globin chains. Isn't that cool? Also, fetuses are sort of parasitic on the mother, but that's a different story we'll come back to later. The hemoglobin molecule has each globin chain has at its center this molecule, which is called a heme group. The heme group has at its center an iron ion, so it's iron 2+. And that iron ion is what actually binds to the oxygen molecule. So since there's one of these heme groups at the middle of each globin molecule, what that means is each hemoglobin molecule binds to 4 oxygen gas molecules. And remember, there are about 280 million hemoglobin molecules in every red blood cell. So each red blood cell can carry 280 million times 4 oxygen molecules. I'm sorry, I didn't do the math on that one. It's a big number. Now, there are cases where hemoglobin does not form correctly. One of those is sickle cell disease. So this is a blood sample from a former student of mine who had sickle cell disease. This is a normal red blood cell. You can see it's nice and round. It looks empty. You can see it's indented in the middle, thicker on the edges. This is a sickled cell that looks narrow and pointy on the ends. Here's another one right there. These here are stuck together because we didn't actually make this slide correctly. Sickle cell is caused by a misshapen hemoglobin molecule. Because the hemoglobin molecules are misshapen, they don't maintain the proper shape of the red blood cell. What happens when someone has a sickle cell attack, which happens often in periods of oxygen stress, such as during exercise or at high altitude, is that instead of their erythrocytes, having this nice round shape and squeezing through the capillaries, the sickled cells actually bunch up and they stick to the walls of the capillaries. They actually start poking into the walls of the capillaries. And so instead of the blood flowing nicely through the capillaries, the blood actually gets stuck. And that can lead to a lack of circulation through certain tissues, and it can lead to death if this happens in the heart or the lungs or the brain. It does cause extreme pain when it happens in other tissues. There is not a lot of research that has gone on until very recently about sickle cell disease, and yes, that is related to the fact that sickle cell disease affects people in the southern hemisphere, especially sub-Saharan Africa. And because sickle cell disease mostly affects Black people, white people haven't researched it. So does racism affect science? Absolutely. It affects science and it affects human health. Now, your red blood cells, your erythrocytes, like all of your blood cells, they are formed in bone marrow, which is these cellular structures inside the spongy bone, inside your bones. We'll come back to that when we do the skeleton. They only circulate for about 120 days in the bloodstream because they don't have a nucleus. Yeah, weird, right? Your liver and especially your spleen are constantly monitoring the red blood cells and looking out for red blood cells that are either old or damaged. And when they find old or damaged red blood cells, special cells in the liver and the spleen phagocytize or engulf those old cells, bind them with their lysosomes and break them down. So we see lots of lysosomes in cells in the liver and the spleen. This is mainly the job of the spleen, but the liver can also do it. So if someone has to have their spleen removed, the liver will take over this job. The components of the red blood cells are going to be broken down and then reused. The heme is converted into a molecule called bilirubin, which is then used to make bile by the liver. And that's an important component of digestion. It's really important for your digestive system. The iron ions, a lot of them are stored in the liver until they are needed by bone marrow for making more red blood cells, and then they'll be released to the bone marrow. And then the other components, all of the lipids, the proteins, the carbohydrates, all of those other things in the cells get recycled by the spleen and the liver, sent back into the bloodstream, and then they can go to other cells to make any kind of new cell or any kind of molecule that other cells need. So that's the life cycle of your red blood cells. Remember that the spleen is really, really important here in keeping your blood healthy. It also stores excess blood. So if you need to increase blood volume in the body, either as a result of blood loss due to an injury, or because you suddenly are running really hard and your skeletal muscles need more blood, your spleen can release more of that blood. So here is a cartoon from the Awkward Yeti, just my favorite cartoon, and I'm going to use a bunch of these throughout these class because they're awesome. Here's spleen saying, I am not beloved, I don't need to be. I filter old blood, I fight bacteria. See, here's erythrocyte and there's a bacteria. I am a silent protector. I am spleen. So be grateful for your spleen, be grateful for your red blood cells. And in the next, we will talk about how the red blood cells are part of blood typing.